Your methodology also involved the Global Statistic Test to combine the cognitive, the functional, and the global outcomes. So, why is relying on a single isolated outcome metric inherently risky and prone to random chance in complex diseases like Alzheimer's?
Okay, so let me use one case that that a lot of Alzheimer's disease researchers will be very familiar with, which is aducanumab. In aducanumab, they had a single primary outcome as their main outcome. It's well known. It's the CDR sum of boxes. This outcome, because there are a few different reasons why it created the controversy, and let just as a reminder, the controversy is that there were two studies. They were two identically designed studies. One is called Engage and one was called Emerge. Engage started a little bit before Emerge, but so they had more subjects that were represented in their interim analysis. In their interim analysis, they had a stopping rule that allowed them to stop for futility. They met that threshold and they stopped the study. And then as the data came in, the results showed that specifically in Emerge for the CDR sum of boxes, there was a positive treatment effect and it was significant. So there's a couple reasons for that. One of those reasons is a statistical one, which is that if you stop for futility, your treatment effect is of necessity deflated. It has been deflated, and it would be more deflated in the study that had a greater representation in that interim analysis, the Engage study. And that shows up in the fact that the CDR sum of boxes was not significant in Engage, but it was in Emerge. The second takeaway from this is that the CDR sum of boxes as a single outcome is more subject to variability because it is not measuring as much of the disease as would be if you had a composite or if you had a global statistical test that is taking into account multiple domains together, hopefully all major domains of the disease, and measuring them at one time. This is again been seen when you look at the other two monoclonal antibodies that have received approval in recent years, lecanemab and donanemab. Both of those in their phase 2 study had their CDR sum of boxes, the one primary outcome that the FDA has seemed willing to use for approval. Both of them, if they had to use the CDR sum of boxes as their outcome that they were making decisions on, both of them failed in their phase 2. However, thankfully, both of those studies, the Trailblazer study for donanemab and the BAN2401 study for lecanemab, both of them are using a composite outcome. With donanemab, it was the IDRIS, and with lecanemab, it was ADCOMS. Both of those composites are broader measures and therefore A, more stable, and B, more likely to pick up disease modification. So because they used those as their primary outcomes in their phase 2 studies, they were able to move on with confidence into a phase 3 study. And thankfully, by the time they ran their phase 3 study, their sample sizes were large enough that CDR sum of boxes actually hit for both of those. And so they had no trouble getting approval based on the CDR sum of boxes with a large sample size. But for many of the Alzheimer's disease researchers and sponsors out there, they don't have the funds in a phase 2 study to use phase 3 size sample sizes. And so if they aren't smart in the outcomes that they choose and make sure that those outcomes are broad enough in what they are measuring, then they are likely going to run into a situation where they are going to fail even if they have an effective treatment.